Welcome to the ACP Problem Solving in Oncology webinar series. My name is Dr. Alison Berner and I'm a member of the ACP Trainees Committee. I'm pleased to be able to welcome you today to our webinar series, which has been set up to provide equal educational opportunities to trainees and fellows in medical and clinical oncology across the UK. Webinars will generally be held on the third Tuesday of each month from 6 to 7:30 p.m. with expert-led presentations and case discussions focused on a particular tumour type or topic. Today's session will cover cancer genomics. There will be two presentations from our expert panellists covering an overview of both Germline and Somatic Genomics, followed by an introduction to Genomics England's Genomes. The educational content of these meetings are entirely non-promotional, but we are proud to have the webinar series sponsored by Pfizer Limited, world-renowned leader in oncology pharmaceuticals. More information is available on their websites. Just to remind you, our online educational platform is delivered by EBN Health. Webinar sessions are recorded and made available to all ACP members via this website. In addition, the website offers access to the ACP's Problem Solving in Oncology book series. To access this, go to www.acplibrary.ebnhealth.com. So I can see that we have a good mix of medical oncology trainees and consultants, and also some of our more junior colleagues on with us today. It's fantastic to have you. And now it's with great pleasure that I introduce our first speaker of the evening, Dr. Terry Mcveigh, consultant clinical geneticist at the Royal Marsden, who specialises in cancer genetics. Dr. Mcveigh is actively involved in a number of trials and educational initiatives. This evening, she will be providing us an overview of germline cancer genomics. Dr. Mcveigh, over to you. Please go ahead and share your screen.
So thanks very much to Alison for the introduction. I'm absolutely delighted to be with you virtually this evening. So as Alison mentioned, I'm going to cover germline cancer genomics. Now, obviously, it's going to be impossible to cover all of the different types of cancers in half an hour. So this is going to be very, very broad strokes, which hopefully you'll still find useful. And if anyone has any burning questions, obviously we'll be having a panel discussion at the end of this session, and I'm very happy to take questions then. In terms of the broad things that we'll cover, we'll just outline very briefly the different types of genes that are involved in cancer predisposition. Not to strike the fear of God into you, but I'm sure you'll remember the nuts and toothpick hypothesis from when you did molecular biology back in med school or more recently. We'll talk about the notion of cancer as a genetic illness, and we'll also obviously focus the vast majority of the talk on heritable predisposition to cancer, particularly considering concepts such as pathogenicity, penetrance, and what do you do when the genetic test is allegedly normal. So hopefully you'll be familiar with the kind of groups of genes that are involved in carcinogenesis. And I always think of it in terms of this kind of fine balancing act between the role of our proto-oncogenes and tumor suppressor genes. So proto-oncogenes are totally normal cellular genes that act to, regulate cell growth and differentiation. And problems arise if you have gain of function or activating variants in these genes. Tumor suppressor genes, on the other hand, work to inhibit replication and proliferation of damaged cells, and they do that by controlling the cell cycle and initiating DNA repair as and when required. And then it follows that loss of function variants in such genes are oncogenic. So I always think about the proto-oncogenes like being the accelerators and the tumor suppressor genes like being the brakes. And if your accelerators aren't working or if your brakes aren't working, it can be equally deleterious. So that balance is disrupted, and that's what prompts cancer to develop. When we think about cancers, we actually need to consider, I've put in inverted commas, two genomes there, because really there's more than two, because obviously cancer is a kind of mini microcosm of multiple sets of clones. But if we think just very broad strokes, we need to consider the genome of the person in whom the cancer develops and the genome of the cancer that they do develop. And obviously if you find a genetic variant in a cancer cell, the difficulty is that you won't immediately be able to determine whether that has been there all along as a potentially heritable event, or whether that has been acquired over time as the cancer has developed. The only way really that you can determine whether it's of germline origin or not is by testing a blood sample to see if the variant in question is also identifiable there. So to draw you back to the Knudsen two-hit hypothesis, Alfred Knudsen was this amazing geneticist. So back in 1971, he looked at a group of children with retinoblastoma, and he divided them up into familial and non-familial groups. So obviously the familial cases had a family history of retinoblastoma, and the apparently sporadic cases did not. And he was able to do this amazing mathematical modelling that helped him kind of work out that this retinoblastoma predisposition had to be related to a single gene, so it was a monogenic disorder, but that actually one variant alone was not enough to prompt cancer development. In order for cancer development to occur, you actually needed to lose the other normal copy of this gene. So we've all got 2 copies of our ORB1 gene. The reason that we have 2 copies of most genes is we get one from our mum and one from our dad. Obviously, if you've got 2 normal copies of the ORB1 gene, your risk of developing retinoblastoma is relatively low. If you're somebody that has a constitutional variant in ORB1, you've lost the function of one of your alleles, but actually your other normal allele can compensate to a degree. And you have normal growth of your retinoblasts. Problems arise if you lose this normal allele. So this is what we call the loss of heterozygosity, or the second hit. And that's really what leads to this loss of control of your tumor suppressor function. And that's what prompts retinoblastoma to develop. And this kind of 2-hit hypothesis is clear in most tumor suppressor genes. And individuals that have an underlying constitutional variant in a cancer susceptibility gene are much more likely to get multifocal cancers, multiple cancers, bilateral cancers, etc., because the likelihood of you acquiring only one additional variant in the normal copy of your gene is a lot more likely than if you're somebody that only has normal copies of the gene and has to acquire 2 separate somatic hits on the 2 copies of the gene. So that's why you get this kind of, they're much more likely to get these things in multiple kind of circumstances and at much earlier age of onset. So if we think just for a second about genetic variation, if you were to sequence the average human being, you would actually find in the order of about 5 million genetic sequence variants. And obviously, the vast majority of us, thankfully, are walking around pretty healthy. So the vast majority of those variants really are just part of normal human diversity. They're just evolution. They account for, the reason we don't all look like clones effectively. But of those 5 million variants, about 10,000 of them will actually change the protein product of the gene in some way. And about 150 of them will actually shorten the protein product. And between 40 and 100 of them have not been inherited from one of your parents. And I'm not getting into non-paternity or anything like that. This is literally just human evolution changing from one generation to the other. Obviously, genetic diversity is a function of time. So you need time for this kind of evolutionary diversity to occur. And it's also a function of your population size. So if you live somewhere like America, where you've got lots of admixture from other places all over the world, they're much more genetically diverse than somewhere like Ireland, which is this lovely little island, way off the west coast of Europe that nobody visits ever, So the island populations that have relatively little inward migration are going to be a lot less genomically diverse than some of the bigger populations. And what that leads to is enrichment of certain variants. You will see these things known as founder mutations that are expressed with a lot more frequently in these smaller populations. The way that we figure out whether a variant is pathogenic or not is not straightforward. So it takes multiple different lines of evidence. So if you sequence somebody's blood and you find they've got a variant in ORB1, for example, you need to use multiple sources of evidence to determine whether it's harmful or not. So you need to look and see, is it present in the normal population? And, if you go out on the street, how many people will you find that has the same variant? If it's very common, it's very unlikely to be something highly deleterious. We also have computational models that help predict what the impact of a particular variant will be on the protein product that's supposed to arise, on the whole process of splicing, etc. We can also do lab studies to literally show what it does to the protein product. And we also use information from segregation. So if you've got 3 sisters with ovarian cancer, do they all share the same BRCA1 variant? If they don't, that's a lot less likely to be the cause of the cancer in that family. Other information as well, so things like, are they inherited from a parent, etc., etc. Has it been seen before in other commercial labs? Is it on ClinVar, etc.? So there's lots of different lines of information that you can use. And luckily, we work with very clever lab scientists who are able to filter down all of that information and categorize variants depending on the likelihood that it is harmful. It's not binary, so it's not a yes or no kind of answer. It's actually 5 different classes depending on the level of suspicion. So if something is, you know, greater than 99% likelihood that it's, you know, causing somebody's cancer, that's deemed pathogenic. Anything between 90 and 99%, that's likely pathogenic. Anything less than 1%, but not less than 0.02% is likely benign, and anything less than 0.2% is benign. And for the eagle-eyed among you, you'll notice then that there's a whole group here of variants that have between 10 and 90% likelihood of being pathogenic, and they're dumped into this category called a variant of uncertain significance. Those variants, so it sounds like we're very kind of city on the fencey about those, but the reason that we put variants into that category is because they don't have enough evidence for us to definitively say in either direction whether they're pathogenic or benign. But more often than not, the reason they go in there is because some of the lines of information are conflicting. We tend to treat variants of uncertain significance as innocent until proven guilty rather than the other way around, because of the possible catastrophic consequences that arise if you misattribute pathogenicity to a particular variant. And there's been lots of legal cases that have arisen because people have been over-eager in, you know, calling a variant of uncertain significance pathogenic. So there was a huge case in the States where a woman found to have a BRCA1 variant and an MLH1 variant, and the surgeon, you know, very enthusiastically did a double mastectomy and a risk-reducing hysterectomy and salpingo oophorectomy. She got millions because, of course, it transpired that they were variants of uncertain significance and she'd had completely unnecessary surgery. So really, we just need a little bit of caution because of the kind of consequences that can arise from it. The other notion that you need to consider when you're talking about variants is this kind of concept of penetrance. So I always think about penetrance very simply, you know, what proportion of carriers of a pathogenic variant are actually likely to develop a disease. And if we think about cancer susceptibility genes and the variants that occur within them, I sort of break them down into 3 groups. So you have, you know, very high risk genes. So if we look here on this, our y-axis here, you can see the effect size attributed to this particular variant, and on the x-axis is how frequently they occur in the general population. These very, very high risk things, thankfully nature is protective, so it you know, means that these things are likely to be extremely rare. Those that are associated with much lower risks tend to be more common because we don't need to be as protective about that. But you can divide your genes into 3 broad groups: high risk that tend to be rare, moderate risk that tend to be pretty rare as well, to be honest, and then these low risk things that are relatively common. If we think for, you know, let's think of a few examples here. If we consider breast cancer predisposition, hopefully everyone in this audience, I'm sure, will be familiar with the BRCA1 and BRCA2 genes, and variants within these genes are generally high risk, and they're pretty rare, although not as rare as we once thought they were. Conversely, then you've got moderate risk genes, or moderate risk alleles, sorry, like variants in things like ATM, CHEK2, etc., etc. The low risk alleles that we talk about, so each of these individually might confer an increased risk of, you know, 1 or 2%, big deal. We're not going to be doing anything drastic on the basis of a single low risk allele. But if you've got 10, 20, 30, 40 of these things, then actually the cumulative risk of that starts to approach that, or maybe even exceed that conferred by moderate or even high risk alleles. And we can generate the cumulative score in the form of a polygenic risk score. And we'll talk about that more later on. If we look at the kind of heritable risk factors for breast cancer that have been identified, there's a serious chunk of it that still has not been explained. Of the risk factors that have been identified, yes, a proportion of them are the high and the moderate risk genes, but actually a significant contribution of the heritability of breast cancer is accounted for by these low penetrance alleles and the polygenic risk score. And that's why we've got these familial clusters of breast cancer that don't necessarily follow a Mendelian kind of pattern of inheritance. The other complicating thing is, although we group the genes into these kind of very, artificial categories, actually you need to think about them in terms of the variants within them. So each individual variant in a particular gene will have its own penetrance. And we know that there are certain variants in BRCA2, for example, that only confer a moderately increased risk of cancer. And the issue with those is that, you know, you may not be as gung-ho about recommending risk-reducing breast surgery in somebody with one of these moderate risk alleles compared to the standard high-risk BRCA2 variants that we're more familiar with. So for these people, it might be more appropriate to offer them surveillance. But again, you just counsel them carefully, and obviously you need to take the patient's wishes into consideration as well. If we think about prostate cancer susceptibility, it's slightly different compared to breast cancer, because you'll notice actually we haven't really identified any very high risk variant alleles for prostate cancer predisposition. We've identified quite a few moderate risk, but actually a lot of prostate cancer heritability remains unexplained, so about 2/3 of it. And of the heritable risk factors that have been identified, the vast majority of them are actually these low penetrance alleles. If we think about it in a slightly different way, if we think about the risks associated, or the risk factors that contribute to certain types of cancers, we know that some cancers are much more likely to have a heritable component than others. And if you look at cancers like breast, colorectal, ovarian, a substantial proportion of that heritability is because of these monogenic high risk issues. But prostate cancer, by far and away, the vast majority of the heritability is accounted for by the polygenic components. We know that other cancers then are less likely to be heritable, and they're much more likely to have environmental risk factors. So obviously, no brainer, you can think of things off the top of your head, things like lung cancer, gastric cancer, etc., etc. They're less likely to have a heritable component, but not impossible. Almost every cancer will have some kind of heritable risk factor. This table, I don't expect you to memorize any of these figures, but it's just to give you a sense of how not rare they actually are. So the average person on the street has a chance of having a BRCA1 variant in the order of about 1 in 380. And I think that's more common certainly than we thought a few years ago. If that person happens to be of Ashkenazi Jewish ancestry, that risk is 1 in 40. If they're a woman that had breast cancer under the age of 40, that goes up to 1 in 8. So they're certainly out there, and they're associated with very, very high risks of cancer. Some of them are much more rare. So if you think about variants in TP53, luckily the prevalence of that is about 1 in 5,000. If we think about STK11, so something like Putz-Jaeger, that's 1 in 25,000 to, as rare as 1 in 280,000 in some patient populations. Lynch syndrome, again, hopefully a lot of you will have heard of Lynch syndrome. It's reckoned now that Lynch syndrome occurs in about 1 in every 270 patients, or 270 people in the general population. So certainly it's underdiagnosed. So you do need to think about it if you have people that have a family or a personal history that fits with that. And again, high risk of certain types of cancers. So when we think about identifying highly penetrant variant alleles, they're what we consider very actionable. And what I mean by that is you can do something to mitigate the risk of cancer in carriers of these highly penetrant variants. So there are guidelines for most of these variants. They're either defined by the UK Cancer Genetics group here. In America, we have the NCCN guidelines, etc., etc. There are some local ESMO guidelines, and broadly, they're all pretty similar by and large. Cancer prevention can involve things like risk-reducing surgery. So obviously for BRCA carriers, that's things like risk-reducing mastectomy, bilateral salpingo-oophorectomy. If we think about chemo prevention in these high-risk people, obviously aspirin has revolutionized the way that we tackle cancer risk in people with Lynch syndrome. We use tamoxifen for people at higher risk of breast cancer. A lot of the kind of risk management hinges around early cancer detection, so with enhanced surveillance. Some of these people will be eligible for personalized treatment. So obviously this audience would be much more familiar with the targeted therapies than I would be. But as well as the kind of systemic treatments, obviously it can influence surgical decision making. So if you've got somebody that's got a bowel cancer related to Lynch, you might do extended surgery compared to somebody that doesn't have an underlying high risk predisposition. Obviously, if you identify a high-risk constitutional variant in somebody with cancer, it doesn't just, it doesn't have implications just for the patient in front of you. It also has massive implications for their wider family, children, siblings, aunts, uncles, cousins, grandparents, second cousins, third cousins. It goes on and on and on. So it can influence quite a substantial number of people. We did a study a couple of years ago where we looked at the number of people in a simple 3-generation pedigree that would be influenced by, finding a variant. In the UK, it averages out at about 17 people. If you're from the Republic of Ireland, it can influence up to 70. So really, family structure has quite a significant impact as well. Obviously, it has implications for family planning. So we're very lucky here in the UK because the NHS will fund options, including pre-implantation genetic diagnosis, whereby the embryos of the carrier and their partner can be tested before it's implanted for a pregnancy, thereby minimizing the likelihood that you're going to, completely removing the likelihood that you're going to pass it on to your progeny. And the vast majority of the highly penetrant cancer susceptibility syndromes have been approved for pre-implantation genetic diagnosis by the Human Fertilization and Embryology Authority. There's also the options of prenatal testing and termination or adoption or gamete donation, etc. So there's lots of options for carriers of high-risk variants. If we think about that group in the middle, so the moderate risk alleles, they're a bit trickier. And if we go back to the situation with breast cancer, you'll remember that we talked about a certain number of genes located in that kind of moderate risk bracket. So things like ATM and CHEK2. They are very tricky to manage, and hopefully this example will give you an idea why. Let's consider this 20-year-old unaffected woman whose mother had breast cancer at the age of 50 years. This green line demonstrates the risk of developing breast cancer in the general population. If we find that her mother has an underlying constitutional BRCA1 variant that accounts for her breast cancer, the daughter's risk of developing breast cancer over her lifetime, if she has not inherited that familial variant, is very, very close to the background population level risk, and we can be very reassuring with her and send her on her way back to population level screening. If mum has a BRCA2 variant that she hasn't inherited, her breast cancer risk is, close to population, but might be slightly increased, not enough to justify any enhanced surveillance. If we start thinking about genes, though, like PALB2, the risk of cancer associated with variants in PALB2 is slightly lower than BRCA1 and BRCA2. So it's kind of lower end of high, upper end of moderate. If that girl doesn't inherit a familial PALB2 variant, you can see, yes, her risk of developing breast cancer is lower than it would be had she inherited the PALB2 variant, but it is still, it's creeping up, in excess of that in the general population. And when we start to consider things like CHEK2, you can see that actually, even if this young girl doesn't inherit the CHEK2 variant that has contributed to her mother's risk of breast cancer, her residual risk of developing breast cancer is actually quite a bit more than the general population level risk. And that's because the CHEK2 variant in her mother likely only accounts for part of her breast cancer risk. It may not fully explain it. And remember, this scenario is a very simplex one. So this is only one case of breast cancer in this family. If you had a very high risk family where, mum, aunt and granny were all affected, then the residual risk might be even higher than that. So this gives us a bit of a tricky situation because the clinical utility of testing this 20-year-old for a moderate risk variant is actually sort of questionable, because if she had a very high risk family history, I'll be offering her enhanced surveillance regardless of whether she's inherited the familial CHEK2 variant or not. So it starts to become very, kind of questionable. If you're not going to change her management, what's the point of offering her the test? And you can see the same kind of situation in somebody with an ATM variant. Her risk is still going to be greater than the population level risk, even if she hasn't inherited it. And that's why, if you look at the NHS directory, a lot of these moderate risk genes are not included on the panels yet, because we're not quite sure what to do with the carriers, but also with the non-carriers in the family. So that's TBD, essentially. So if we think about guidelines for moderate risk alleles, mostly they're not there yet because we're still kind of debating in the background what we should be doing with them. There's some guidelines from the States, primarily the NCCN, but they're quite heavy-handed, possibly compared to what we would offer in NHS clinics. In terms of cancer prevention, so risk-reducing surgery is not typically recommended for carriers of moderate risk genes or variants. So if you think about risk-reducing mastectomy, we sort of draw a line in the sand at 30%, and if your risk of developing breast cancer over your lifetime is not greater than 30%, you're very unlikely to qualify for risk-reducing mastectomy. Similarly, if your risk of ovarian cancer is not greater than 10%, we're unlikely to offer you risk-reducing salpingo-oophorectomy. In terms of chemo prevention, it's all about risk-benefit analysis. So like, if you're a young woman that has a 25% risk of breast cancer, but you're also predisposed to developing DVTs, maybe tamoxifen is not the right thing for you to be on. So it's all about looking at the patient in front of you and taking all of those factors into consideration. Surveillance, certainly there's a case to be made for it, but the surveillance that we offer to women at moderately increased risk of breast cancer is nowhere near as high intensity as it would be for somebody like a BRCA carrier. So you're mainly focusing on mammographic-based screening rather than MRI. In terms of personalized treatment, possibly, maybe, there's some positive studies out there looking at PARP inhibitors and ATM variants, things like that, but that's all still sort of to be clarified. Cascade testing in the family, as I said, there's a big question mark over the utility of that. So that's again to be determined. Family planning options. This is really contentious because the moderate risk alleles are not necessarily likely to be HFEA approved or funded. The difficulty with some of these is that some of these variants some of these genes are implicated in other disorders, so they may appear on for another reason. So for example, ATM is associated with another disorder called ataxia-telangiectasia, which is a horrible neurologic disorder that occurs in kids that inherit 2 copies of an ATM variant. So there's all a bit of kind of greyness around that. And there's ongoing discussions between the UK Fetal Genetics Group and the UK Cancer Genetics Group to kind of come up with sort of an agreement about what level of risk would justify doing pre-implantation or prenatal testing. And again, that's not an easy question to answer. Let's focus a little bit on the technical approaches to identifying these kind of gene variants, because obviously we're in generation genome. The whole genome test is hurtling rapidly towards being available for diagnostic testing pretty routinely. In the very simplest kind of way of doing genetic testing, you can literally look for specific variants. So, hopefully this audience are very familiar with the BRAF V600E variant that occurs in melanoma and in other cancer types. If you wanted, you could literally just look for that particular variant, but you're kind of limiting the utility of your test because you could look more broadly for other variants in that particular gene. You could focus literally on that gene, or you could focus on the gene and the other members of that particular family. But actually more and more, we're increasingly using things like whole exome, where you sequence literally all of the coding regions of the known disease genes, or whole genome, where you sequence not only the coding regions, but also the non-coding regions as well. In reality, actually, whole exome and whole genome are just kind of fancier panels, because the way that you do your analysis is you kind of virtually filter out your genes of interest. So essentially, we consider multi-gene panels, whole exome and whole genome, sort of along the same lines. The higher up those steps you go, the more data you get. So certainly you increase your likelihood of finding an actionable genetic variant. You also increase your likelihood of finding incidental variants in genes that might not necessarily be causative, genes that might not necessarily be even moderate risk. And certainly you increase the likelihood that you'll find variants of uncertain significance, which can cause massive headaches. If you don't find any variant, I am very, very slow to ever call a gene result normal, because every test has limitations. If you have a family like this example here, sorry the pedigree is so small, but that woman there had breast cancer, her mum had breast cancer, maternal aunt had ovarian cancer, another maternal aunt had breast cancer, another maternal aunt had breast cancer, 2 cousins with breast cancer, does not take a geneticist to look at that pedigree and go, oh my God, there's something going on here. But all of her high risk gene tests were negative. In this family, there is clearly some kind of heritable predisposition that we just have not identified. So I would, go back to this woman and say, you haven't identified a variant in your family, doesn't exclude a heritable risk factor. I still think you should have enhanced screening. So if you're suspicious, and the things that make you suspicious, are there lots of people diagnosed on the same side of the family? Are they diagnosed at very young ages? Or are there other suspicious features? So if you had 3 sisters that all happened to have triple negative breast cancer, obviously you'd be a lot more suspicious than if you had, you know, 3 sisters that had ER positive breast cancer in their 60s. Certainly things like mismatch repair deficiency, if there's examples of that, that needs to prompt consideration for Lynch syndrome in your head as well. So you can offer enhanced surveillance, and actually in some situations you might offer risk reducing surgery if it hits those kind of estimated thresholds that I talked about earlier. The kind of limitations of the testing that I referred to there, so if you think about your panel-based test or your exomes, you're not going to pick up variants in the intronic regions and the non-coding regions. You're not going to pick up structural rearrangements. So there are these things known as translocations, whereby material is swapped from one chromosome to another. I always think about your genetic information. If you think about your genes like being words in books, chromosomes are like the shelves that contain those books. And when you're looking for mistakes in words, you're not going to know if one shelf has been moved from one part of the bookshelf to another. So it won't pick up that because it's not looking for it. It won't pick up methylation defects where the genes totally normal in terms of their sequence, but they've been silenced, they've been switched off. So it won't pick up that. Won't pick up other things like triplet repeat disorders. And there are some regions of the genome which will literally just be very difficult technically to sequence. So if you ever look at a gene test report, you'll always see the little rider at the bottom that says this is the limitation of detection, 99%, etc., etc. Like with any test in medicine, you know, it has limitations. Even genome, apparently the panacea of, the cure to all of our ailments, there are certain variants that it will not detect either. So it can detect intronic variants and it can potentially detect structural rearrangements, but actually that's very contingent on your technical approach and your bioinformatic pipeline. And I have seen, particularly through the 100,000 Genome Project, I have seen instances where these have not been detected because the manner in which the analysis was undertaken. And similarly, you have technical issues with your whole genome analysis as well. So if you have a genotype negative family, negative does not necessarily mean normal, doesn't necessarily mean not genetic. You need to consider what genes have been tested. So if you have tested a woman for BRCA1, BRCA2, PALB2, that doesn't necessarily exclude the rarer syndromic causes of breast cancer. So you always need to think of those as well. Ask your local friendly geneticist for an opinion if you're very worried. You need to think of the limitations of the test you've undertaken, and you need to consider alternative mechanisms of disease. So, there's epigenetic, there's potentially the polygenic risk scores. There is also the notion of shared environmental influences as well. So, we see some families with clustering of lung cancer, and actually when you ask them, all of them have been smoking 40 a day for 20 years. So, there are shared risk factors that are not necessarily heritable. Polygenic risk scores, as I said at the outset of this talk, polygenic risk score is effectively a quantification of the cumulative impact of a number of low penetrance alleles. Most of these are single nucleotide polymorphisms, so changes in a single nucleotide. Sometimes within a gene, but actually for the most part outside of genes, so in intragenic regions. There's hundreds of SNPs that have been identified for cancer. If we think about prostate cancer, for example, there's about 200 of them have been identified so far, of which a proportion are associated primarily with aggressive and early onset disease. If you generate a polygenic risk score, what it gives you is a relative risk compared to the specific patient population in which it was created. And of course, like an awful lot of genetic research, this, derivation of polygenic risk scores have largely been based, they're sort of Eurocentric. So that, application of a PRS score generated in a European population may not be applicable for somebody from Southeast Asia, for example. So it's influenced heavily by ethnicity. You're very unlikely to get an absolute risk estimate from a polygenic risk score. It'll just say you've got a risk score in the top 10th centile. Your relative risk is 2 or 3 times the background level. So this again is the prostate polygenic risk scores, and you can see this middle region here is your baseline, and if you're in the top 1%, your relative risk of developing prostate cancer is 5.7-fold. If you're in the lowest 1 centile, then your relative risk is 0.15. And we're using this, so there are studies ongoing in the ICR that are looking at the utility of screening using PSA, MRI and biopsy in men in the top 10% of risk, and we'll see what happens with that study. In terms of actionability, this is very question mark, because there's no guidelines. Cancer prevention, again, it's all under investigation at the minute. We're not at the stage at all yet where we'd be recommending risk reducing surgery, because we just don't know enough about how well PRS performs in different contexts. Similarly, chemo prevention, we don't necessarily recommend on the basis of PRS alone. There are studies that are looking at the influence of polygenic risk scores in terms of lifestyle modifications. So there's the Lioness study here in the Marsden that's looking at that. Early cancer detection, again, surveillance, that study that I mentioned, the ICR looking at PSA-based surveillance, potentially, it'll be very interesting to see what comes out of that. Personalised treatment, that's unlikely because these aren't, you know, targetable variants at present. Cascade testing in the family is a nightmare because the polygenic risk score, remember, might be comprised of 130 individual SNPs. So, you know, the son of a father that has X number out of those 130, he's only going to have a 50-50 chance of inheriting each of these individual components. So the likelihood of him inheriting the same set of 130 as his dad is 1/2 to the power of 130. So incredibly small likelihood of that. So it's not inherited in the strict 50-50 sense, the way that BRCA variants are, for example. Family planning options, no way, like there's not, it's just not technically possible at the minute to offer pre-implantation or prenatal testing for polygenic risk. Watch this space, so who knows what will happen in the next 20 years. So just in summary, sorry if I'm gone slightly over time, what you need to consider when you're thinking about constitutional variation, you know, should I be doing a germline test in this person? You know, are they young? Have they got a weird phenotype? Have they got a really strong family history? What implication is it going to have for their treatment? Is it going to make them eligible for immunotherapy? Is it going to make them eligible for a PARP inhibitor, etc.? You also need to think about what you're going to do with the result. So am I going to be able to interpret what a variant in this gene means? You know, is the variant pathogenic? Is it in a gene for which guidelines exist? What am I going to do in terms of risk reduction for the person in whom it's identified, but also for their sister, their brother, their son? You need to think about family planning considerations. So what are they going to do when they want to have their own kids? And you do need to remember the limitations of each of these tests. And if you can remember only one thing from this talk, remember that a negative test does not necessarily exclude heritable risks. If I ever see any of you write not genetic in a letter, I will come after you. So I think we're very familiar with this whole notion of the right test, the right patient, the right time in medicine, and the same applies to genetics as it does to everything else. So hopefully that's been useful, and I'm happy to take questions at the end.
That was fantastic. Thank you so much. And the not genetic in a letter, I will keep that well in mind and spread the word to my colleagues. I think we have time for one question that's in the chat at the moment from Sarah, I think. Are genetic services streamlined across the UK? i.e., is there equal access to tests, geneticists, no matter where the patient is?
Yes, technically, I think we're getting there. So this, the NHS directory has, so there's been this reconfiguration of NHS services, which has centralised all of the testing. So germline testing now is being carried out in hub laboratories, and we're all supposed to follow the NHS directory, which has set out kind of criteria that we should all be following now for testing. So, you know, in the Marsden, we used to be free and easy with genetic testing. If you walked up and down the corridor often enough, you'd get a genetic test of some sort. And we've been brought back down to earth, and we're not allowed to do as much. But places where it used to be much more restrictive, now they have much more in the way of access to it. So it is supposed to be becoming kind of more standardized across the board. I think that has taken a bit of time, and it is still continuing to take a bit of time. The rollout that's really been quite slow is mainstreaming. So, you know, here in the Marsden and other parts of London and in Southampton as well, where Dr. Copson is, you know, oncologists are doing an awful lot of their own germline genetic testing, whereas I know other places, oncologists haven't been able to do as much of that, but hopefully that will be, you know, coming on stream very, very quickly. And I think that's why there's initiatives like Genomes and like these webinars where we want to upskill everybody and bring everyone up to the stage, you know, so that everyone is able to do it in the same way. So hopefully, I think probably not right this minute, but hopefully in the very near future, it will be equitable across the board, yeah.
Following on from that, there can be quite a lot of heterogeneity in even the reports that we get sending off genetic testing. Will the advent of the genomic lab hubs bring that into a standardized format as well?
Yeah, and so the really lovely thing about working in the UK at the minute, it's, you know, sorry, this sounds really cheesy, but it's really exciting to be working in genomic medicine in the UK at the minute, because there's so many collaborative groups at the minute. So, you know, the ACGS, which is the Association of Clinical Genetic Scientists, they've got amazing guidelines. They're trying to come up with standardization for literally not even the wording, but also the template, so that if you get a report down in Southampton, it'll nearly be the same kind of report that I get up in London. And the wording, so the interpretation, you know, what does pathogenic mean? What does that mean in terms of diagnosing this person? So you'll see, if it says pathogenic, it'll say confirms a diagnosis of Lynch syndrome. If it's likely pathogenic, it'll be consistent with the diagnosis of Lynch syndrome. If it's a VUS, it'll say hold your horses, don't do anything, this is not actionable, you know. So it is, I think all of that is coming, and there's been an awful lot of work, thankfully, in trying to make this all very patient friendly, because what we want to do is get to the stage where we're giving the patient the report, and if it's patient friendly, it'll be doctor friendly, you know. So it'll be really kind of, you know, obviously with the greatest respect to everybody, but not everybody is as genomically literate as some other people, so we're trying to make it so that it's very user friendly for everybody, so you'll know immediately what to do with the result, and you won't be going out and doing unnecessary mastectomies on people. Well, I'd hope the average medical oncologist wouldn't be doing that anyway, but you know, that it's trying to safeguard against that over-interpretation that has happened in some of the legal cases previously. So I think the next couple of years we'll see a lot of standardization across the board.
That's fantastic. And I will just squeeze in one more question from Pauline Leonard. Good to have you on the webinar. She's written, now I'm worried about reassuring patients with a family history of colorectal cancer whose tumour is proficient in MMR. What should I be thinking or saying to that individual?
Oh, that's a really good question, and I'd love to start talking about colorectal, and Alison is equally obsessed with colorectal cancer, so we could talk about it all day. Colorectal cancer is a really tricky one because, of course, the incidence, particularly in the Western world, is increasing at, you know, a horrible speed, and you do get this familial clustering. Like, certainly Lynch syndrome by far and away is the most common heritable cause of colorectal cancer, and if you've, you know, effectively outruled that to the best of your ability by IHC or MSI testing, you know, if they have a family history that still looks very convincing for Lynch, so if there's lots of colorectal cancer and there's a couple of endometrials, I would still do the genetic testing, because MMR IHC is only about 96% sensitive. So there is a case to be made for those families, and actually they're eligible under the directory. If they've got that kind of family history, you should still do germline testing for that. If it doesn't look very Lynchy, you know, the other kind of subset of colorectal cancer predisposition syndromes are the polyposis. If they don't have any polyposis, it's unlikely to be one of those conditions. But, you know, again, some of them will qualify for that polyposis panel. It may well be that it's one of these colorectal cancer syndrome X type scenarios that we don't know about. So, you know, they will still potentially need a bit of enhanced screening, and the BSG have excellent guidelines about that. And, you know, it's now I need things spelled out to me, so it's very clear as well about how many relatives affected and what the surveillance should be for those people as well. So I can forward that paper onto you, Alison, and maybe you can share it with Pauline if that would be helpful.
Brilliant. Thank you very much.
Great.
Now, moving forward, it's with great pleasure that I introduce our second speaker of the evening, Dr. Ellen Copson, consultant academic medical oncologist at Southampton who specialises in breast cancer. Dr. Copson is actively involved in clinical trials, including 100,000 genomes, and is cancer lead for the Wessex Genomic Medicine Centre. This evening, she'll be providing us with an overview of somatic cancer genomics. Dr. Copson, over to you. Please go ahead and share your screen.
So my job is, over the next 25 minutes or so, is to give you some insights into somatic genomics as opposed to germline. And, you know, like Terry, there's no way I can tell you about every somatic test and how you should use it to treat your cancer patients, particularly because I've only treated breast cancer patients for the last 10 years or so. But what I'm going to try and do is just sort of give you a bit of an insight into what's sort of changing from an NHS point of view. And in the second half of my talk, to give you a bit of an insight into what I've learned, particularly in my role as co-chair of our regional molecular genomics board over the last 5 years, and the sort of lessons I've learned when it comes to interpreting somatic genomic tests. Okay, so what I'll basically be covering is the evolution of precision oncology, new NHS genomic services, and lessons learned. So you will all be very, very aware that the idea of precision oncology is really not a new one. And of course, by that we mean identifying the treatments that are going to most benefit an individual patient based on a real understanding of the molecular biology of their personal cancer, in a way that we can hopefully define the treatments that are most likely to succeed, and in particular avoid the treatments that are most likely to fail. We've been doing this for a long time. You know, we've been using trastuzumab in the breast cancer setting for 20 odd years now. But historically, we were very much looking at protein markers, either on the outside of the cell through immunohistochemistry, or by using in situ hybridization technology to look at sort of macroscopic changes in chromosomes to identify particular biomarkers. And what's really changed over the last 20 years is that increasingly we are looking at the actual sequence of the DNA, or sometimes the RNA, within cancer cells. And we're now using that genomic information both in the context of predictive and prognostic biomarkers, but even it's starting to sort of make its way into monitoring. Now, in the interest of time, I'm going to very much stick to talking about predictive biomarkers today. And the reason that we are able to use genomic markers now in the clinical setting are because of the huge advances in technology. That means that whereas the first human genome took 12 years to sequence, we can now do it in a couple of days, just over £1,000. So it's actually, you know, this technology is now bringing this sort of cutting edge science right into the clinic room with our patients. We know that there are lots of different types of genomic aberrations that we see in tumours, as well as point mutations, small insertions and deletions. We can see much more extensive changes, so structural rearrangements. And the sort of key thing about the driver genomic changes we see in cancer cells is that they tend to upset the really key biological pathways that are involved in cell proliferation and survival and metastatic spread. And, you know, it's really, you know, and a lot of you are too young to remember a world before some of these treatments. When I started my oncology training exactly 20 years ago, and in those days, nobody wanted to do melanoma because, you know, it was a choice between dacarbazine and best supportive care. And of course, the advent of the BRAF inhibitors, such as vemurafenib, which was very specifically designed to target an altered protein that was found in a significant proportion of melanoma patients. And, you know, the key biomarker for that, of course, is the V600E mutations. And we've all seen the data from the pivotal clinical trials that brought this into routine practice. And we've seen the effects as well in terms of the, you know, a rapid improvement in disease-associated symptoms that many patients treated with this drug receive. Similarly, lung cancer, you know, back in the day, it was a choice, you know, were you going to give carboplatin and vinorabine, or carboplatin and gemcitabine was the big decision to make about your non-small cell lung cancer patients. And of course, we now have a host of targeted therapies, such as the gefitinib and the GFR inhibitors, and more recently the ALK inhibitors as well. So genomic information really has sort of transformed the way we are treating patients in our clinic. The latest sort of kid on the block is the NTRK inhibitors. And just to make sure that some of you out there are still awake and listening and haven't just put your headphones down and gotten to watch EastEnders, there's a question for you, and we should be able to get a poll up. So which of the following tumour types are the NTRK inhibitors approved in now by NICE? Excellent. So it looks like that the vast majority have heard of these drugs, which is fantastic. So these are targeted therapy that have been heralded in the sort of public press as being the first sort of tumor agnostic treatment. So NTRK fusion genes are oncogenic drivers of a whole variety of different pediatric and adult tumors. And there are now a couple of drugs now that have been specifically designed to inhibit that particularly abnormal protein. And what's interesting about these fusion genes is that they're found in very different frequencies, but in a wide variety of different tumours. So hence the phrase agnostic tumour therapy. But these drugs are now approved for treatment for a large number of solid tumours, providing the disease is locally advanced or metastatic, and they have this, NTRK fusion biomarker, and they have no other satisfactory treatment options. Okay, so sorry, here we go. So if you look now at any list of targeted therapies, you will see a sort of a whole range of drugs. And although some of them are still reliant on immunohistochemistry testing, increasingly we'll see a move towards genomic biomarkers as being the sort of key eligibility feature to be able to access these drugs for your patients. The big exception, of course, is immunotherapy, which is very much reliant on immunohistochemistry. And of course, within immunotherapy, we're seeing a sort of a whole range of different tests, different antibodies, different thresholds provided, in different settings. And this is, exactly what we saw historically with all the genomic tests too. They were usually reliant on the pharmacological company coming out with a companion test when they were trying to persuade us to sort of start using these drugs. Then as their oncologists started asking for the test, regional genetics labs would kind of see that this was kind of quite a good area to get some business from other places, and they would set up block contracts. So what happened historically is you had genomic tests being performed all over the place, different laboratories, some commercial, some NHS, different techniques, and often different results as well. And because of that, it has been a bit of an unsustainable situation, particularly as more targeted therapies have come into the market. We've had this sort of slightly mad situation where certain tumour samples have had to do a swift tour of the country to go to several different laboratories in order to have the different tests. The funding streams have been sort of opaque at best and non-sustainable at worst. But perhaps key in terms of patient care, what there has been historically is huge variations in the use of genomic tests in different cancer centres and in different parts of the country. And that's translated to a lack of genomic knowledge in parts of the clinical community as well. So over the last 10 years, the NHS has embarked on a really ambitious agenda to get genomics at the forefront of medical care in this country, particularly cancer care, to make sure that we're all genomics aware, and to make sure that our patients all have access to the same test in a timely fashion, regardless of where they're being treated. And the sort of first sort of marker on the route to this was the announcement of the 100,000 Genomes project. David Cameron, whatever happened to him, he announced this in 2012, and it was this incredibly ambitious project which aimed, as the name suggests, to sequence full genomes of 100,000 cases. And this was a combination of rare diseases and cancer cases. And it was all delivered via a network of 13 NHS genomic medicine centres working with local hospitals. And the whole idea was that it would sort of be paralleled by research developments in a university education in genomics as well. And the sort of theoretical pathway that patients went into this was on paper quite simple. Essentially, you had your cancer patient who was going to either have a biopsy or surgical removal of their tumour. We would extract DNA from that and from a germline blood sample. That DNA would then be sent to a big warehouse outside Milton Keynes, where whole genome sequencing of both the tumour and the germline blood sample would be performed. We would then be sent back the results, and perhaps really uniquely to this project, we then had a responsibility to inform the patient of the results as well. I think in principle, it was a lot more challenging than at first sight. And, you know, a lot of the lessons I've learned have been, you know, as a result of my work within this project. And I'm going to share those with you shortly. The next thing that sort of happened on the route to sort of genomic equality was the NHS set out a big project to look at exactly what tests were being used where, and it made a commissioning sort of commitment that every genomic test that was cited in a NICE technology appraisal would be then sort of centrally funded from that time onwards. Interesting, HER2 and EGFR have always been excluded from this arrangement because they were sort of deemed to be routine practice by the time this was put in place. And then the third step along the pathway was the opening of the National Genomic Medicine Service in October 2018. And this now consists of 7 regional genomic laboratory hubs, which are coordinating a network of subcontracted local genomic laboratories. And that network is, you know, completely responsible for all genomic testing now and going forward. We now have 7 genomic medicine service alliances sort of alongside those which are sort of in parallel working on the clinical aspects of genomic medicine. And all of this is sort of looked over by NHS England. And what is happening in terms of cancer care is that we are, there's sort of a plan to move from the immunohistochemistry single gene tests and the sort of small to medium sized panels that have been, you know, sort of our common practice, and, you know, the sort of aim is that eventually we will get to sort of much larger scale cancer panel analysis coordinated by the laboratory hubs. That is very much a work in progress. You know, it will depend where you are at the moment as to exactly what tests are being done on your tumours, and, you know, at what rate those are going to change to larger panels, but they, you know, they can be accessed in general at the moment. And perhaps sort of a key part of this whole program is that whole genome sequencing is now available for selected indications. And at the moment, those are selected hematological malignancies, pediatric tumours, and perhaps most relevant to the audience I suspect is there tonight, is that sarcomas are now eligible for whole genome sequencing. Please don't start sending samples in without discussion with your local genomics team, because there are certain pathways and processes that have to be you know, taken into consideration here. And in particular, you will need to complete a record of discussion, you know, and you need training to know how to do that before you can submit samples for this. And there are very clear pathways that have to be gone down. And exactly as Terry was just explaining, perhaps the really key change for us and our pathology colleagues now is that tests have to be within the scope of the National Test Directory. We can't just do what we like on who we like. You know, we have to, you know, stick to the National Genomic Test Directory. And there is a separate directory from rare diseases for cancer. Cancer susceptibility germline tests are all within the rare disease genomic test directory. Somatic tests are within the National Cancer Genomic Directory, but even within that, there's a bit of a mix. So if you go onto a tab for any particular tumor type, you will then also sort of find a mix of predictive genomic tests, and you will also find some prognostic tests in there, some tests to help you with diagnosis, for example, of obscure sarcomas. And you will also find in somatic testing, germline DYPD testing. So, you know, it's a little bit of a mix there at the moment, but it's really worth looking at for your tumor type so you can kind of see where things are going in terms of somatic genomic testing. Okay, so I'm going to move on now and spend, you know, the last sort of 10 minutes or so just sharing with you some of my experiences and you know, the things I think it's worth being aware of when you are doing more and more genomic tests, and in particular, you know, talking to patients about genomic tests. And so just before we dive into that, I've got another quick test to check you're all still with me. Can I just have your answers to this one, please? The vast majority of you will have got this, but there are a few people who are going for the wrong answer here. So hopefully my next two slides will clarify this. When we are talking about somatic mutations, we are talking about the changes within cancer cells. And those are by definition, in their own right, never heritable. You will have somatic mutations in patients with a genetic cancer susceptibility syndrome, but those somatic mutations, you know, on the face of it, are not inheritable and not inherited and can't be inherited by your offspring. Okay, so this is lesson one for you. Language really matters, because if you're talking about genetic testing or genomic testing, you know, it's really unlikely that the patient in front of you understands what you are talking about unless you explain it a bit to them. As Terry said, you know, germline genome is what we've inherited and it's what we can pass on, and that might include all sorts of cancer susceptibility mutations and all sorts of mutations that influence our risk of getting other diseases. But when we're talking about cancer genomics, the somatic genome changes, we are not going to pass those on to offspring. They are not in the germline genome. And so what I often hear in my clinics is patients will say to me, you know, have you done genetic testing on my tumor? And you know, sometimes that's because they you know, have heard of Oncotype, which is obviously you know, also a genomic test, but will tell me whether or not I should be thinking of adjuvant chemotherapy for a patient. But I always now say to the patient, you know, that's an interesting question. What do you mean by that? And quite often this is the reply I get, so that I know whether my daughter is at risk of breast cancer. And so immediately you've got people who have heard about genetic testing or genomic testing. They assume it's been done on their tumor, but what they think it's going to tell them is something about their genetic susceptibility to cancer, and therefore their child's genetic susceptibility to cancer. So it's really important that you unpick that, and if you're doing genomic tests, that you explain to the patient what you're doing, what information is going to give you, and what implication that has for the patient and for their family, so that they're really clear about this. It's equally important when you're talking to other specialists as well. I reckon about 90% of the time that oncologists talk about genomic testing, they're talking about somatic testing, but I reckon about 90% of the time geneticists are talking about genomic testing, they're talking about germline testing. It's really important we understand what the other person's saying. And, you know, it causes issues, it causes distress to patients if they misunderstand it, it causes confusion amongst the, you know, the MDT if everyone's talking about different things. So it's really important that you're clear. We've had situations locally where we have had lung cancer patients being referred into the genetics team because their oncologist told them that they had an EGFR mutation, and they were very, very worried about passing this on to their families. So they went to their GP, and their GP referred them to clinical genetics. And obviously, A, that's not a good use of anyone's time, but B, you know, it's causing unnecessary concern and distress for the patient. So please be really clear about what you're talking about if you're doing genomic testing or if you're talking about the results. My second lesson was that whole genome sequencing may be sometimes just a bit too much of a good thing. And as Terry has really nicely described to you, you know, whole genome sequencing, gene panel testing, they're all sort of variations on the same theme. They're also using the same technology. But with whole genome sequencing of a tumor sample, we're not going in with sort of very preconceived ideas of where we think the hotspots are where we want to pick up, which is what happens in gene panel testing. And it does have, you know, key differences in the way we go about it. Firstly, for whole genome sequencing, you need more DNA, you need higher quality DNA. And what you'll end up, I always think of it icing a cake. If you've got a really big cake, you're going to end up with quite a thin layer of icing, where if you have lots of smaller cupcakes, you can sort of pile the icing on quite thick. So you get much sort of deeper coverage when you're looking at gene panel testing. And this is why sometimes you'll actually miss things with whole genome testing that gene panel testing has picked up. And the attractions and for whole genome sequencing is that you can pick up stuff you won't get on you know, some panel tests. For example, tumor mutational burden we know can be a biomarker for response to immunotherapy. It saves you doing lots of different tests on one tumor. You're just doing it all in one go. And from a technical point of view, you know, the argument you'll often hear, well, it's value for money. And, you know, it probably is if you're just looking at the whole genome sequencing. The issue is there's quite a lot of manpower required before and afterwards. So I would say the jury's out on how really good value for money it is. And there are some key challenges, and you need to be aware of this, particularly if you're in the sarcoma field and want to access whole genome sequencing for your patients. So first of all, formalin fixing damages nucleic acid, and what we saw in the 100,000 Genomes project was that we got much poorer results of whole genome sequencing from formalin fixed samples than those that were fresh frozen. So we need fresh tissue to do NHS whole genome sequencing. That has logistical issues. It means your biopsy or your tumor sample's got to get straight to the path lab, and it needs to get there and somebody be ready to deal with it, because it's going to require expert dissection to preserve your sample integrity and to assess tumor cellularity, because all that information is needed as well. The other big thing is that in order to interpret your tumor whole genome sequence, the gold standard is to subtract the germline sequence. And that is what is being done in NHS whole genome sequencing. And that means whether you were really planning to send your patient for germline genomic testing, you're going to get it anyway. And this is why there is this process or recorded discussion, because if you're doing NHS whole genome sequencing, you are going to get germline information, whether you particularly were after it or not. And that has implications clearly for the patient, but also for their family as well. And, you know, interpretation, because there is so much data, because there is germline data you weren't necessarily looking for, it takes time and expertise to analyze, and this is where tumor boards have really sort of come into their own. And you know, it does cause problems. So we had a patient, 77-year-old, who went into whole genome sequencing. She had her lung cancer resected, and then she went off and was discharged from you know, surgical follow-up, and as far as we can tell, is doing very well. She had no history of breast cancer or ovarian cancer, but a germline BRCA2 mutation was found on her whole germline whole genome sequence. And when we tried to tell the patient about this, they wanted nothing whatsoever to do with it. They won't come to clinical genetics. We've spent a lot of time discussing how to manage this, because the big question is now is, do we as doctors have any ethical responsibility to other family members? And that is a sort of situation that is still under discussion at the moment. So, you know, once upon a time we used to do tests because we wanted specific answers and we knew what we were going to do with those answers. But actually, when you go into whole genome territory, you might get a lot of information that you weren't necessarily looking for, and then you have to try and work out what to do with the results. Okay, lesson 3. Somatic testing does not take the place of germline. So about 5 to 7% of breast tumours will have what looks like a pathogenic somatic BRCA1/2 mutation. And most of those will have an underlying germline mutation, which is relevant to future cancer risk and to their family, but not all of them. Also, because of the nature of germline pathogenic BRCA1 and 2 mutations, often they're large deletions, they won't all be detected by somatic sequencing approaches. So they are really important to understand that somatic and germline testing are not equivalent. You can't tell somebody they've got a germline mutation until you've actually looked for it. Okay, lesson 4. And that sort of comes on from that really. Not all mutations are equal. So the way Genomics England have structured their whole genomic sequencing reports is that they talk about domain 1, 2, and 3 variants. Domain 1 variants are variants in a virtual panel of potentially actionable genes. And these are defined as genes in which small variants have been reported to have therapeutic, prognostic, or clinical trial associations. Okay, so they are being categorized at a gene level rather than a variant level. So the sort of situation we would come up against in 100,000 genomes, was a lung cancer case, an EGFR mutation would be flagged up to us as an actionable mutation. But when we actually looked at it in detail, it was outside exons 18 to 21, which is the tyrosine kinase domain. It wasn't in the cosmic database of, actionable variants. And the clinical utility of this is really not established. And although that had automatically put in a hyperlink for trials of EGFR mutation inhibitors, when we actually looked at the eligibility criteria of the trials, this particular variant didn't meet those eligibility criteria. So this is again where a tumor board really comes in helpful with the whole genome sequencing, because you really need to look in depth at the variants you're being told about. And it's worth just being aware that the ASCO, in conjunction with the American Molecular Pathology Board, has come up with some consensus guidelines which are slightly different, and they tier variants at variant level rather than gene level in terms of the evidence base for their clinical significance. So if you are sort of getting genomic information from other sources, you may well see it presented as a tier rather than a domain result. Okay, lesson 5. I promise there are only 7, and I'll sort of race through the last few because I know the evening's coming to an end. But so clinical contact is important. I think most of you will know that BRAF mutations and BRAF inhibitors have been really valuable in melanoma, but not in colorectal cancer. Very limited response there to BRAF inhibitors on their own. But what's looking slightly more promising there is BRAF or sort of other in EGFR inhibitors, MEK inhibitors in a sort of combination fashion. And similarly, in the breast cancer environment, we've seen that AKT inhibitors have some clinical benefit in certain settings, but interestingly, in triple-negative breast cancer, the presence of an alteration in the PTEN, PIK3CA or AKT pathway does seem to predict a greater response. In ER-positive disease, the presence of that biomarker really doesn't seem to impact on the clinical benefit. And again, it all seems to be because of interactions with other clinical elements, such as here ER status also comes into play. Okay, penultimate lesson, and you'll all be familiar with this one, and something I say a lot more to non-oncologists is that clinical utility is not the same as actionable mutation. And actionable mutation, there are lots of different definitions. At its simplest, it's about predicting response to targeted therapies. It can be used more broadly. It can be used as a genetic level rather than a variant level, as I just explained. Clinical utility is whether you can do anything with it. And I was sort of really always the harbinger of doom in our molecular tumor boards, because our fantastic biomedical scientists would have spent ages trying to find me an actionable mutation, and then I would have to tell them that was nothing I could do with it because that drug isn't NICE approved, or the patient had too many lines of therapy already. And this is a really important issue, particularly if a patient's talking about self-paying for genomic tests, because they really do need to understand that, you know, just because a report says there's something actionable, it doesn't actually mean the oncologist is going to be able to act on that. And the last lesson, which again, as oncologists, you will be familiar with, but I have spent a lot of time trying to explain to non-oncologists over the last few years, is that genomic biomarkers, as with all biomarkers we use, they predict response, but they certainly don't guarantee it. And actually, I came across this recently, which was a really nice summary of response rates of targeted therapies with biomarkers. And you can see there on the slide on the left that we're generally looking around a sort of 40–50% objective response rate, which, you know, essentially means in clinic, it means half the patients you put on a drug are going to benefit from it. So really important that we again, coming back to my first lesson, we use the right language, and we talk about, you know, the becoming eligible for treatment, or, you know, hopefully benefiting, but you can't guarantee it. And, you know, of course, our issue in advanced cancer is the heterogeneity of metastases, the evolution of metastases compared to your primary tumour, which means even if you identify a biomarker in one bit of cancer, it doesn't mean all the cancer is going to respond to it. And, you know, I'm sure you will all have seen this sort of follow-up photo to the photo I showed earlier, which sort of, I think, you know, really does demonstrate, you know, that resistance to targeted therapy is a very real issue. We are starting to identify markers of resistance, and for example, the T790M mutation, exon 20, is now recognized as occurring in about 50% of cases of acquired resistance to EGFR TKI therapy, and that test is available for non-small cell lung cancer within the new Genomics directory, so we can start to sort of interrogate this idea of markers of resistance. In personal point of view, I think a lot of the sort of future of genomic work, and in particular tracking those evolving changes in our tumours, is going to be aided by all the work that's been done in the area of liquid biopsies. So that's actually doing blood tests to assess the genomic sequence of circulating cell-free DNA. And if I recommend one genomics paper for your bedtime reading, it's got to be Nick Turner's paper on the results from the plasma match study, which was an umbrella study of metastatic breast cancer, with a number of sort of different therapies matched to various sort of genomics finding and the circulating cell-free tumor DNA. And it's a really nice paper, which I think really highlights the complexities, but also the potential of this sort of approach. So I'm going to stop there. I hope what I've got across to you is that, you know, we are all increasingly and going to be even more so in the future using somatic information to inform our cancer treatment decisions. Whole genome sequencing is now available within the NHS for those selected cancer patients, so that's sarcoma patients in terms of adult tumours. But I think it is, you know, important for you to sort of understand the challenges as well as the benefits of this approach. Presence of a genomic target, as I said, it certainly doesn't guarantee activity or availability, and it's just important to be aware of that when you're talking to your patients, but also to non-oncologists as well. And circulating tumor DNA, I think, represents a really exciting method of identifying genomic alterations and monitoring them through a patient's journey. I'm going to stop there, but thanks very much for listening.
Thanks very much, Ellen, for a fantastic talk. This is such a fast-moving field, and there's so much to cover. So you've done a fantastic job in whizzing through it in that time. And there's some brilliant discussion going on in the chat as well. So a chat between again, a great question from Pauline Leonard, answered by Terry, and with myself chipping in around repeat testing for MMR, and whether we're seeing MMR proficient tumours develop, say for example, MSH6 mutations after chemotherapy, and whether we would then think about offering immunotherapy as the phenotype changes, or whether we wouldn't confidently call that, because it's unlikely to be at a particularly high frequency within the tumour, and that's just an example of loss through tumour evolution.
We were talking about this. Sorry, we have a hereditary bowel meeting with St. Mark's and the other amazing experts in the Polyposis Centre, and the quality assurance program obviously, you know, looks at the kind of calling of MMR, IHC and other IHC generally among the pathology labs, but actually it's incredibly assay dependent, and it's very operator dependent as well. So if you ever have like discordant results, sometimes I will ask it to be repeated, you know, because sometimes it could just be actually a mistake in the test rather than a change in the tumour. You know, there's limitations to everything. You know? And
I think perhaps this is where one of those times when whole genome sequencing does help you, because I know from my PhD work that actually it's a completely different phenotype across the whole genome with regards, and the signatures would really help you out there, and that may be one of the utilities of moving towards.
And actually even your morphology. So like if you've got a tumour that's got a shed load of tumour infiltrating lymphocytes, and it's mucinous, and it's got all of those kind of features of mismatch repair deficiency, sometimes you don't need to do any test. That's a kind of proxy, you know, pseudo marker that it probably is immuno driven, you know. So
there's another great question. So Sarah had mentioned, would we be doing more extended germline testing and extended somatic testing coming through, and actually Peter Johnson's picked up that that's going to produce quite a large volume of work for the genomic MDTs, and is there perhaps a role for artificial intelligence or some other mechanism to streamline this process?
I hope so. I really hope so. So, I mean, certainly what we'll be doing locally is that whole genome sequencing results will come through our molecular tumor board, but we certainly don't envisage, you know, going through all the panel tests or large and small, unless they've sort of picked up particular concerns or, you know, particular cases that they want us to review. I think one of the key things with all of this is, you know, essentially is how do you set your filters, and at the end of the day it's all about, you know, the sort of software, which results you pull out. And I suppose my slight, I hope there aren't any radiologists on the call, but my kind of perception of radiology reports over the sort of last 20 years is that once they kind of used to be really clear, and they tell you things if they were definitely there, but if they weren't quite, you know, if things were a little bit on the fence, they wouldn't necessarily mention them, whereas now they tell you absolutely everything and say, you know, this may not be clinically significant, but we're going to tell you about it just in case. And I think this is the where we sort of are at the moment with a lot of these genomic reports, is that, and the way the software is set, is they'll want to tell us every single thing just in case it's clinically useful, and then we sort of find ourselves in the situation, as we do with radiology, you know, trying to do tests and biopsies to kind of, you know, reassure everyone that it's not clinically significant. You know, we sort of go down that same rabbit hole with some of this genomic information, and I think it's you know, all going to be about setting the filters at a point where we can be really confident that this is positive and clinically useful genomic information, rather than telling us all the different, you know, information just in case. I think the problem is, of course, that it's all moving, and, you know, with 100,000 genomes, we got reports being reissued, you know, time after time.
Yeah.
We kept going back to them to see, you know, if anything had changed, and inevitably that is going to happen. So it's having those sort of workable mechanisms in place there. You know, if we are really, really convinced there's something really interesting going on in this family, and there must be something somewhere, you know, we can kind of reaccess the information as the evidence base increases, but actually so that the amount of information we're getting through on a sort of regular fashion is manageable. And I think this is, as I say, you know, a few years ago I was told by NHS England that I would be doing whole genome sequencing on all my breast cancer patients within a year, whether I wanted to or not, and I'm really relieved. They haven't, they came back on that, and at the moment it's just my sarcoma you know, colleagues that we're sort of needing to support from and as well as paediatric and hematological.
One final question before we move to the Genomes demo. We know that there's DPD testing now routinely done. Are there any other pharmacogenomic tests on the horizon that you're aware of?
So Munir Pir Mohammed is doing an awful lot of work in this space, and he's going to be advising the NHS about implementation of pharmacogenomic testing, because obviously it's not just oncology, there's implications for anaesthetics and warfarin and clopidogrel and everything. So the difficulty with a lot of the pharmacogenetics is that actually the effect size is tiny. So if you think about some of the anaesthetic variants, like it might mean that you take 25 seconds longer to come around from your anaesthetic than, you know, so some of the effects are not massive. So I think Munir hopefully will guide all that. That's a very tricky area, you know.
Thank you very much. So in the last couple of minutes, I would just like to demo something that myself, Ellen, Terry, and some other colleagues have been working on. This is a platform called Genomes. It's being done in the as a collaborative effort between HEE, the Academy of Medical Royal Colleges, and various specialties, including oncology. And it's actually about to move into its phase of alpha testing. So if you're able to see my screen, so this is what the platform will look like, and it's supposed to be a just-in-time resource. So the purpose really is when you're in the clinic and you're faced with a particular clinical scenario where it would help you to have some additional genomic knowledge, you would be able to go to your relevant specialty, and you can go through and find a number of clinical scenarios. There'll of course be many more than these, and find a scenario that matches your situation and find more information. And so clicking on the individualized patient case, you would be able to have a look through the scenario, find some background information, and then click links out to resources. So for example, here you have the National Test Directory. And then moving back into it, you can also move into further, more in-depth information on areas of interest. So for example, here, more information on Lynch Syndrome. And those, what we call tier 2 resources, are actually contained in a knowledge hub where you can actually go through and just pick a topic where you're not coming to information that's driven by a particular clinical situation and you're just looking to learn more. And so, as I said, this is moving into a phase of alpha testing, and we're looking for both consultants and specialty doctors to test the platform as part of that alpha phase, which is coming through in May. And if you're interested to take part, then I'd ask you to please contact the email on the screen, which is info@lagomstrategy.net, and put Genomes in the subject line. So we're not allowed to give out emails, but if you provide them your email, you have early access to the platform for your own learning and be able to test it as part of the alpha phase. The beta phase will be coming through shortly in August, and I can ask the ACP to send out further details around the alpha and beta testing phases in due course. So thank you all once again for attending this evening's ACP Problem Solving in Oncology webinar. We can only provide certificates to those who complete feedback, so please do remember to do this for us. There's also a space to enter suggestions for further topics in the future, which we would very much encourage. Our next session will cover acute oncology in the UK, and that will take place on Tuesday the 18th of May at our usual time of 6 p.m. And this is a special joint session which will be delivered with the Royal College of Radiologists, with a multidisciplinary panel including both clinical and medical oncologists discussing the state of AOS in the UK, their experiences and its inclusion in the new oncology curricula and the challenges that lie ahead. You can register now at www.theacp.org.uk, and on behalf of the speakers and everyone at ACP, thanks again and good night.